My name's Vikas Bansal. I'm from UC San Diego, and I'll be talking about low mappability genes in the human genome and what challenges that presents for variant calling and a solution for this problem. Some background on human genome sequencing. Last 15, 17 years, we have tremendous advancements in DNA sequencing technologies, and Illumina is the dominant technology for at least sequencing human genomes. We have now long read sequencing, but they are much more costly. Illumina generates short read lengths. Everyone probably already knows that. And whole genome sequencing costs about $600 for a human genome at about 30x coverage. And as a result, we have several, by some estimates, million genomes sequenced since 2008. And there are public databases available that have information from some of these genomes in the form of variants and their frequencies. Since Illumina sequencing generates short reads, so the typical approach for at least analyzing DNA sequencing data from human genomes is to do variant calling. And what is variant calling? Variant calling is basically a computational process of comparing the sequence genome in the form of short reads to a reference genome to identify positions in the sequence genome that are different. And here's kind of a very high level overview of how variant calling works. So you have a reference genome shown on top here, just a small section of the reference genome. And on the bottom, you have the aligned reads that are also sorted by position. So each gray bar corresponds to a aligned read. And you can see that there are certain positions where the aligned reads, one position here, half of the aligned reads indicate the presence of a variant because they have a different nucleotide or multiple nucleotides different than the reference genome. So that's at the high level, that's what variant calling is. It seems simple, but there are a lot of challenges associated with sequencing errors, alignment errors, and so on. And this is like, it's been, a lot of tools are available for variant calling, and it's very, very accurate for at least single nucleotide variants. One of the challenges for variant calling comes from the fact that the human genome is highly repetitive. So there are, in the morning talk by Powell, we heard about centromeres and also about these specific loci like the IGV locus that are highly repetitive. And so in addition to that, there's about 7% of the human genome that corresponds to segmental duplications or low copy repeats. So what are segmental duplications? So first defined back in 2001, these are sequences in the genome that have high sequence similarity to at least one other sequence in the genome, and also are at least 1 kilobase pair long. So that's kind of the definition that's been used over the years. And this figure here is basically a chromosomal snapshot of segmental duplications shown in the highlighted by the red bars. And you can see they are all over the genome. Some chromosomes have a higher density, others have a lower density, but they are ubiquitous in our genome. And so that poses problems for variant calling because some of these segmental duplications also overlap genes, and we care about genes in the context of human disease. So about in 2016, a group looked at the impact of these segmental duplications on variant calling using short reads. And so what they found was that about 2,000 genes in the human genome have regions that are have overlap segmental duplications or other types of repeats that are problematic for variant calling using Illumina reads. And they also found that 50% of these genes are covered, they are not just 1 or 2 exons that are problematic, but more than 75% of the exons for these genes have low mappability. And so, and many of these genes are already linked to human diseases. Some of them were mapped before Illumina and short read sequencing were developed. So clearly, variant calling is problematic for a significant proportion of genes in the human genome. And to kind of illustrate the computational problem that this poses, here's an example of a gene called nebulin. This is a large gene that is known to mutation in this gene cause a rare disease called Nemelin myopathy. And this gene has this interesting kind of duplication where 8 exons in the middle of the gene are repeated 3 times. So you have a 10 kb sequence that covers these 8 exons, and in tandem you have 3 copies of this 8 exon sequence. So obviously you would imagine that variant read mapping in this region would be problematic, and so here I'm showing you the alignment of the reads in the form of an IGV plot. So again, the gray bars, which may not be visible, correspond to the aligned reads, and you have the 3 red boxes correspond to the 3 different copies of this triplicated region. And on the right hand side is the same IGV plot, but only the reads with high mapping quality or reads that can be mapped with high confidence are shown. So you can see that in the triplicated region, we are left with a very small fraction of the reads. That's not surprising because reads that originate from these 3 copies, they cannot be mapped uniquely because each read has potentially 3 different alignment locations. But standard variant calling methods rely on mapping quality to call variants. So because if a read cannot be mapped with high quality confidence, then you cannot use that for variant calling. So as a result, in this region, with standard variant calling, variants cannot be called. So kind of to go a little bit deeper into the how read mapping works in these duplicated regions, I'm showing you an example of a 2-copy duplicated region. So you have copy 1 and copy 2. These 2 sequences are highly identical, and I'm showing you one position specifically that differs between the 2 copies. So these positions are known as PSVs or paralogous sequence variants. So that's a terminology that's been around for a long time, but you can just imagine them as positions that differ between the different repeat copies. And these are important because in these regions, these are the only positions that can tell us whether a read came from copy 1 or copy 2. So for example, if I have a read that has the A sequence at this position, then I'm maybe confident that this read came from copy 1. And if it has a G, then probably more likely copy 2. So essentially, read mapping in these long duplicated sequences relies essentially on PSVs. So the more the PSVs you have, which means you have low sequence similarity, it's easier for the Illumina reads to be mapped to the right copy. Whereas if you have very low number of PSVs, then you cannot really map any reads uniquely. And so this concept of mapping quality, again, mapping qualities are basically a Fred scale probabilities value. So 0 would mean that you have 0 confidence or that the read has been mapped to the correct location, and higher values indicate that you are more and more confident that you have mapped the read to the correct location. So there is another twist in this problem is that even in regions of duplicated regions that have low sequence similarity and a lot of PSVs, you cannot always trust the PSVs to distinguish between the repeat copies. And the reason for that is that gene conversion is a mechanism that can result in copying DNA sequence from one repeat copy to the other copy due to the fact that when these repeat copies are proximal, and that can result in the PSV turning into a polymorphism. So in this example, we have 2 PSVs, this A corresponding to the G and then T and C. So if gene conversion happens and copies over a small stretch of DNA from this C allele over to this copy, then this individual A doesn't have the T, but the C allele at this position. Now, when we sequence the genome of this individual, any read that came from copy 1 but had the C allele would incorrectly be mapped by a standard read mapper to this location with high confidence, or at least moderate confidence. Because the reference genome tells us that this position, if it has a C, then it's copy 2. And so as you can see, that will result in incorrect read mapping, which can lead to incorrect, false positives and false negatives during variant calling. And this gene conversion is actually a very common phenomenon in many low mappability genes that have the two copies like located close to each other. So that's just another kind of showing you illustration of what happens when reads that are incorrectly mapped with high mapping quality to the incorrect location, that can result in the variant being called at the wrong position. And so you have a false negative because you didn't call this variant here, and then you have a false positive. Another example of a challenging case for a standard variant calling is that when you have no PSVs, and so all the reads have low mapping quality, and so basically then you end up with false negatives. So in this case, standard variant calling cannot do much because the reads have low mapping quality, and kind of a hybrid situation where some of the reads overlapping a variant have high mapping quality because they happen to overlap a PSV on the one side, whereas the other some reads don't have low mapping quality depending upon where the PSVs and the variant positions are. And so this kind of mixed situation can cause also incorrect variant calling and genotyping. So kind of another complication when it comes to low mappability genes is that they also tend to vary a lot in copy number. So they are actually the most polymorphic, in terms of copy number in the human genome. A study published by Evan Eichler in 2010 showed that about 40% of the genes of these multi-copy genes that have low mappability have common copy number variation. And why is this problematic for variant calling? Because again, variant calling assumes that you have diploid, so you have two copies of every chromosome, every region. But for example, if you have in this case 3 copies of this sequence, then not only is your genotyping going to be incorrect because you are trying to fit the read counts observed from the 3 copies onto a diploid genotype. So kind of to summarize, variant calling in low mappability genes, there are like 3 different challenges that I've tried to kind of given you a little bit of overview. So first one is kind of more obvious that mappability is a problem, but some of the other ones are not so commonly recognized. The fact that PSVs can actually cause incorrect read mapping due to because some of the PSVs represent polymorphisms, and also the fact that copy number variation is not accounted for when doing variant calling. So actually for copy number estimation, there are existing methods out there that can estimate copy number even for these low mappability genes using short read data. And this was first shown back in 2010 by Evan Eichler and his group, that you can actually estimate both aggregate. In this case, aggregate would mean that how many copies of the different repeat copies you have in total. So in this case, you have 3, copy 1, and then copy 2, a total of 3 different repeat copies. And then parallel specific copy number would mean that you have 2 copies of the blue one and 1 copy of the other one. So, and then this was done in 2010. So a few years ago, we developed a method that is a targeted method. So it can, given any specific low mappability gene, it can analyze just that specific gene without doing any genome-wide analysis to estimate aggregate and parallel specific copy number. So copy number information can be recovered for these low mappability genes. So what about the other 2 problems? So let's go back to the example that we had earlier for the nebulin gene, where we have this triplicated region where a lot of the reads have low mapping quality. So now one possibility is that why can't we do variant calling on all the reads, like just assume that all reads have high mapping quality. Well, the problem is that if there is a variant present in one of the copies, then because the read mapper assigns reads randomly to the different repeat copies, if it has equal, equal kind of alignment score, then the reads that have the variant, they will not be present on a single location. They will be distributed across the 3 different copies.
Does that still hold if you make the aligner align in each location it can instead of randomly choosing one?
Yes, that does. Yeah, if you do like a exhaust, like all versus all, then you will have that. But again, we are working. If you're looking at standard alignment tools, that's not the standard like the default mode. The default mode is to align each read to exactly one location and assign the mapping quality. So in this case, a simple solution is to basically uh, mask out the extra repeat copies. So in this case, copy 2 and 3, and then align the reads, realign the reads back to this kind of modified reference genome. And what will that do is that all the reads that were aligned to copy 2 and copy 3 will now be aligned to copy 1, and they will have high mapping quality because now because we masked out copy 2 and 3, there's only one location for these reads. And as a result, you can still, and also the problem that all the reads, as you mentioned, that have the variant in a specific location, they will all be piled up in the same position. So but now the challenge is that this region of the genome is not deployed anymore. So because we've aligned the reads from these 2 other locations to this single region, now this region actually has a ploidy of 6. And so we have to do variant calling accounting for this higher ploidy, and then hopefully we can identify variants. So so that's kind of the basis of this approach that we developed a couple of years ago. So multi-locus approach for variant calling in these low mappability genes using whole genome sequencing, and it basically combines kind of these ideas to solve those 3 different challenges that I mentioned earlier. And I'll kind of go over this algorithm in detail to give you a sense of how it works. So the first step is actually what we just talked about earlier, which is to kind of remap the reads from all the other repeat copies to a single repeat copy. So hopefully that makes sense to everyone why we do that. So that basically allows us to discover the variants, because now all the reads for a specific position across the different repeat copies are going to be piled up in a single location. And also now we don't have to worry about mapping quality or any errors in the original mapping. And thus, the assumption is that there are no other regions of the genome that have the reads, from which the reads could also be mapped to this location, and that kind of is implicit because we consider all other similar sequences in the genome for kind of remapping the reads from those regions to this single region. Once the reads have been mapped to a single repeat copy, we can identify variants using a standard variant calling tool. We use Freebase because it allows for providing or specifying the ploidy. In this case, the ploidy would be the aggregate copy number, which we can already estimate using the tools that I talked about earlier. So once we do that, we have variant positions shown here and also the genotypes. And the genotypes are slightly different than the diploid genotypes. These have higher ploidy basically, and they correspond to the genotype of a specific position aggregated across multiple repeat copies. And so to give you a little bit more detail, what are aggregate and paralog specific genotypes? So again, going back to our example of you have 2 copies of the same kind of sequence, high sequence similarity. These are 2 paralogous positions between the repeat copies. Now, if an input genome has A and G on the 2 chromosomes in copy 1 and A on here, then the paralog specific genotype is A/G for copy 1 and A/A for copy 2, whereas the aggregate genotype is just kind of the concatenation of these two genotypes. I hope that's clear to everyone. And so what we are doing is first we identify variant positions, estimate aggregate genotypes, and once we have the aggregate genotype, all that remains is to kind of split it back into the parallel specific genotypes. And then obviously there are multiple choices, and we select the one that gives the best likelihood. Okay. So going back to the next step in the algorithm, which is to identify reliable PSVs. So remember I mentioned earlier that not all PSVs can be used to, should be used to distinguish between the different repeat copies, because some PSVs can actually correspond to polymorphisms. So how do we identify reliable PSVs? Well, there are two criteria. One is that in the individual that we are looking at, in that person's genome, the aggregate genotype for that PSV should match the reference genotype. So in this case, the aggregate genotype is two T alleles and two C alleles, and this matches the reference genome because the reference genome you have expect 2 copies of a T and 2 copies of C. And also the PSV allele frequencies, which I didn't talk about so far, which basically tell us something about at the population level how variable are the genotypes at this PSV. So PSV allele frequencies basically represent kind of like a summary of what is the allele frequency of each allele in the different locations of a PSV. So for example, I have this one PSV with the A allele here and a G allele here. If I looked at in the population, then 100% of individuals have A in copy 1 and G in copy 2. So I don't see any, like G allele in copy 1 and vice versa. Whereas if I look at PSV2, then in copy 1 we have 100% T, but in copy 2 there is a mix of C and T. And I won't go into the details of how we can estimate these frequencies, but that's part of the copy number estimation. So for this talk, we'll just assume that we have these frequencies, and they allow us to identify PSVs that can be used for distinguishing the repeat copies. So in this case, this PSV is considered reliable, whereas this PSV, because the genotype for this is not the same as the reference, so it actually has 3 A's, so which means that there is a polymorphism at this position, at least on one of the 2 locations of the PSV. So this PSV here is reliable. So now we look at the aggregate genotypes for this first variant. This is 3 A's and a G. Now what that means is that the G allele has to be present on either copy 1 or copy 2. So that's all we have to determine in order to resolve this aggregate genotype into parallel specific genotypes. And how do we do that? So let's say we have a read that has a paired end read that overlaps the variant and this reliable PSV. So this paired end read has a G allele for the variant and a T allele for the PSV. So what that means is that the G allele must be present on copy 1, right? Because that's if a G allele is present in copy 1, then only you can see a read with G and T together. So kind of like phasing, but slightly different. And that's if we have multiple reads, we can combine the evidence from that, and basically that resolves this aggregate genotype into this parallel specific genotype. Similarly, we can do the same for variant 2. Variant 2 has 2 G's and 2 T's. There are many more options for resolving that aggregate genotype into parallel specific genotypes. But because the T alleles are always observed along with the C allele for PSV1, we can confidently resolve it into G/G and T/T. And in the case of this third variant, because the neighboring PSV is not reliable, we are unable to resolve this variant into parallel specific genotypes. So, but for this variant, we still know that there is a variant present, but we do not know whether the variant is present in copy 1 or copy 2. And actually, these variants are still useful because, for example, in the case of the NEB gene I showed you earlier, it doesn't really matter whether the variant is present in the 3 copies because they're all exonic. As long as it's exonic and a missense, it's still likely to be potentially disease causing. So having information about aggregate genotypes and variants is still useful for downstream analysis. So this is the only kind of the results I'm going to show in this talk. So we benchmark this method, this multi-locus variant calling method, using genome in a bottle benchmark data for this one genome that had long read based variant calls for some of these duplicated low mappability genes. And what we found is that compared to these standard, this 3 different standard variant calling methods, this multi-locus approach does better on precision and recall. And as the sequence similarity increases, then our method has a greater improvement in both precision and recall. And this is not surprising because regions that have very high sequence similarity, that's where you would expect this problem of low mappability to be the most challenging for standard variant calling. So basically, think I'm at the end of my time. So just to summarize, I gave you like these kind of somewhat new concepts of aggregated variant calling, parallel specific genotyping, reliable PSVs that are not many of you probably have heard or seen before. And these are the key kind of the ingredients for this multi-locus method for variant calling in low mappability genes. And a useful feature of our method is that it's a targeted method. So if you just want to analyze a specific disease gene in, let's say, 100,000 genomes, you can do that efficiently with this approach compared to some of the previously published methods that kind of require whole genome remapping or reanalysis. And the references that were listed on the program website, they're actually missing the couple of the references that are relevant to what I talked about today. So if you're interested, you can take a look. And I just want to close with acknowledgements. Timofey Prodanov, he led the development of this multi-locus variant calling method. He's now a postdoc at Heinrich Heine University in Germany. And then another student in the lab is working on further developing this method for whole genome and whole exome data. And I also want to acknowledge the NHGRI for funding this work. And I'm happy to take any questions.